My name is Scott Soefje. I'm Director of Cancer Care Pharmacy at the Mayo Clinic in Rochester, Minnesota. And today I want to talk a little bit about biosimilars. In 1984, Congress passed the Hatch-Waxman Act, which established generic drugs. And it really gave us the first look at what we can do to lower the drug costs and really improve the cost of care for patients. Um, and as we began to look at biosimilars, it became obvious we needed something from the biosimilar perspective, or biologics perspective, because biologics are continuing to grow, and next year are projected to surpass 300 and almost $380 million worth of costs, and we believe biosimilars can have a very significant impact on that. So in 2009, as part of the Affordable Care Act, the Biologics Price Competition and Innovation Act was passed, and it was designed to mimic the Hatch-Waxman Act, in able to establish an abbreviated process for biosimilars to be approved, and really begins to set up a process for biosimilars to be treated similar to generic drugs, even to the point where they established a purple book, which talks about the biosimilars and whether they're interchangeable or not interchangeable and all those kind of things. So when we talk about biosimilars, we have to think about biologics and small molecules as different. Biosimilars are close to generics, but they're not the same. Um, the size of the molecules are different, the way things are produced are different, and so biosimilars will never be an exact copy. Whereas a small molecule, you can actually make exact copies. Now, we also have to remember that from batch to batch, all biologics, even the brand names, are not exact copies of each other's. There's a variance that they're allowed to be in, and that's something we're going to talk about in a second. So really, to understand biosimilars, pharmacists really need to understand how biosimilars are approved. And if you look at this graph and you think about the reference product, when that first product comes out, we really don't care about the structure and function. Yeah, it's nice to know, in some of the early clinical studies and mechanism of action and all that, we want to know what the mechanism of action is, but how it determined is not always interesting. PK/PD becomes a little more interesting for pharmacists. We want to know, how long the drug's going to be around, what it's going to do, that kind of stuff. But the real focus is on the clinical studies. Does this drug do what they think it's going to do? Does it do what they say it's going to do? However, biosimilars flips this model on its head. And if you think about it, what really becomes important from a biosimilar perspective is the structure and function, is the mechanism of action working the same, and are we getting the same responses from a PK/PD perspective? When you put all that together, you can say these drugs are doing the same thing in the body. They're binding and working through the same mechanism of action. Therefore, maybe the clinical studies don't mean as much. And all we need to show is that there is some activity. And that's how biosimilars are set up to approve. And so what happens is, the the act established this process to become something that's highly similar, meaning there's no clinically meaningful differences in safety, purity, or potency. And in the legislative language, potency means activity. And so what the FDA does is they start with the analytical component, which is the bottom of the pyramid. They move up to the non-clinical data. They move up to the PK, PD, and immunogenicity data. And if there's uncertainty at any level, they make them go to the next step, to the point where the FDA could truly approve a biosimilar on PK, PD, and immunogenicity if they felt that they had demonstrated that the mechanism of action was strong enough, et cetera. In most instances, what we're seeing is at least one or two clinical trials that are being done in sensitive patient populations to make sure that there's activity. When we look at the structure and function, these are the different aspects that the FDA is looking at. And you can tell it's a significant amount, very heavily molecular chemistry and biochemistry to make sure that these molecules are the same. And in many instances, the amino acid sequences are identical between the biosimilar and the reference products. Where you start seeing changes is in all of the other things, the process impurities, or maybe some glycosylation profiling and stuff. The question is, does that make a difference in how the drug is acting? As I said before, there's this normalized range that every biologic, whether it's the reference product from batch to batch, or whether it's the biosimilar from biosimilar to reference, has to be within this normalized range. If it's in this normalized range, then the FDA considers them the same drug, particularly on the reference product, and they consider them biosimilar if they are on the biosimilar side. So then we have to understand the concepts of extrapolation. Extrapolation then says, if the evidence of the biosimilar suggests that it works in one area, in that one clinical trial, and the mechanism of action is the same, and the PK/PD is the same, and the toxicities and immunogenicity is the same, then we can say that we can extrapolate that this drug will work in other indications as well. That's how biosimilars get around with only doing one clinical trial versus multiple clinical trials to get their drug approved. An example. So Neupogen's indications were approved. These are the indications in the box. When the drug Nevastim, which is a biosimilar, came across, they did a single PK/PD study in multiple healthy volunteers. And what they were able to show is they got white cell growth, because that's what the drug is supposed to do. They did a non-inferiority study to show that the immunogenicity and safety in that population was the same. So the FDA said that there was no clinically meaningful differences and gave Nevastim the exact same indications as Neupogen, except for, if you'll notice, the last indication is an orphan indication, and that's protected by legislation. So Nevastim doesn't have the orphan indication. Otherwise, the indications are exactly the same. And we see this with all biosimilars. And this is what you hear. Sometimes you'll hear it called a skinny label, because the biologic doesn't have the exact same, or the biosimilar doesn't have the exact same as the reference product. Interchangeability then takes this a step further, this whole concept. And it means that you could switch the products back and forth, just like we do with generic drugs on a daily basis, and there would be no efficacy difference, no safety difference, no concern. And when a drug gets interchangeability status, it can truly be treated like a generic drug. The pharmacist can change it out without talking to providers. Uh, Otherwise, the pharmacist has to follow their state laws on whatever their laws require on biosimilars. We're starting to see interchangeable drugs, particularly in the Humira market. We're seeing them in the insulin market. We're seeing them. So we're going to see more and more interchangeability starting to occur as we move forward. Whether it's necessary or not, it all depends. Some institutions allow therapeutic substitution, so interchangeability may not be required. Again, interchangeability just adds that next layer, but it requires the pharmaceutical company, the biosimilar company, do that extra clinical work to show that these drugs can be switched back and forth with no problems. So again, if you're thinking about how the drugs are approved, we go through the analytical PK/PD. We show that from the analytical perspective, everything's the same, the mechanism works. Then we get an extrapolation so that the indications don't have to be clinically tested all the time. And then if the company takes it to the next step, they can get interchangeability, which means that switching back and forth between these products is not a problem. If we do this right, biosimilars can have tremendous impacts on clinical practice. If we have widespread adoption by providers with a growing familiarity and a comfort level, then what we can see is that we can see a decrease in the cost of care, and it will lead us to decreased overall costs. Right now, payers are driving the decisions on which biosimilars, because they're getting tremendous rebates and tremendous cost savings from the pharmaceutical companies. What we'd like to see is biosimilar parity and allow us just to choose which one's the cheapest, because we believe, just like generic drugs, these products work across the board. And ultimately, what we're trying to do is lower the cost of care for patients, lower their out-of-pocket expenses, and decrease their overall cost of care without compromising care. So I hope you learned something about biosimilars today, and I appreciate your attention and your tuning into these discussions.